these, excitatory postsynaptics is going to be caused by the particular type of neurotransmitter and its receptor that is going to cause either a depolarization or a hyperpolarization, right? Because these inhibitory neurons are going to secrete a certain type of neurotransmitter onto a postsynaptic membrane that has a certain type of receptor, right? That's going to be different from this and this. And so this is what we're getting into next, these kind of channels that are on the postsynaptic membrane and then the actual neurotransmitters that cause these effects. In general, there are two general classes of neurotransmitters, receptors. So here we're talking about receptors are these ionotropic and metatropic, right? This is what we've been talking about most of the time, right? This is what we're talking about with our acetylcholine. In fact, here's the example of acetylcholine, which it's a ligand-gated channel. When the neurotransmitter, in this case, the acetylcholine, this is just an example. It could be other ones. When it binds to the channel itself, the protein itself opens up, and then whatever it's going to allow through it goes through it based on their concentration gradients, right? So sodium and calcium are going to flow in the cell in this case, and maybe potassium is going to flow out. Whatever the channel is, some are going to only allow sodium, some are only going to allow potassium, but the binding site and the channel is in the same protein. The other type, a little more rare and a little more complicated, gets more confusing, is these metabotropic receptors. And in this case, the channel here, the receptor that's binding the protein, the transmitter, in this case, it's again acetylcholine. What does that mean? We'll talk about it in a second, but in this case, the binding of the neurotransmitter doesn't open up anything in itself, but rather it's coupled to G proteins, which, if you'll remember, was one of the ways when we talked about hormones, right? That class of hormones which bound to receptors and caused this G protein and that cascade of second messengers and so on, right? In this case, the most easy thing to think about is that this G protein over here that's bound to this receptor, once activated, it causes somehow, magically, don't worry how, it causes other channels to open, right? So it's going to be coupled to another receptor, which is going to cause another channel to open. And this way you can modify the whole signal, as we'll see. Those are the difference between these two basic types of receptors. This doesn't tell us anything about excitatory, inhibitory, so let's go to that. Most of the things we talked about so far, right, were excitatory one. And we mentioned many times sodium flows in because sodium was highly concentrated on the outside. Calcium was highly concentrated on the outside in the extracellular fluid. So when these channels open up, calcium would rush in right here, right, causing a depolarization because this was negative, now it's positive, right? That would be influx of a positively charged, a cation, right, coming in causing depolarization. Or other types of neurotransmitters are going to open up, say, a chlorine channel, which is the main anion out in the extracellular fluid. Remember, sodium and chloride are floating out there, very high concentrations outside, very low in here. So when you open up this chloride channel, the chloride ions are going to rush in the cell over here, and that's going to make the cell even more negative, right? Again, from -70 to -90, for instance. And that would be an inhibitory postsynaptic potential. That would make the cell, if there was an adjacent synapse nearby that was excitatory, that cell would have to work harder now because now the cell is going to be harder to reach a threshold. It's going to diminish. It's going to kind of negate that whole excitatory input right there. Right? So these are two examples of ionotropic, and that's the basic thing, right? Either positive ions, mostly sodium for the most part, and the other one will be chlorine. Both of those are coming inside the cell because they're highly concentrated, outside the cell. So here's another one, this metabotropic center, when we're talking about inhibitory ones. When the neurotransmitter binds to the metabotropic receptor, the G protein is going to directly open or close that channel. And in this case, we're talking about a potassium channel. Right? And this potassium channel opens up. What happens? Where's potassium? Potassium is highly concentrated inside the cell, so it's going to rush out. Potassium is positively charged. What happens when potassium leaves the cell? Right? It makes it more negative inside the cell because this was a positive charge. And it's leaving, according to its concentration gradient. But this is a metabotropic, and it's, one of the ones where we're going to use potassium instead, an outflux of potassium, right? The other ones were influx of sodium or chloride, bringing either positive or negative charge. And this one is a potassium one leaving the cell, which is going to create this negative charge. And those are your basic types of flow of ions going into or out of the cell right here. And some examples of how they might do that. You may have noticed during all this that both of these were acetylcholine. So acetylcholine was being released into the, this is not specifically, I'm not really specifically talking about acetylcholine, just the fact that in one case over here, the binding of acetylcholine opened up this channel, and there was a net influx of positive ions. Right, all these guys came in and all of a sudden made this more positive over here. And that was due to acetylcholine. This is exactly, again, what we saw with the neuromuscular junction. On the other hand, if you were at a synapse that had these types of acetylcholine receptors, when acetylcholine binds, it would do that thing we just talked about right there, right? It would open up these potassium channels instead, because the receptors were different. One was purple, one was yellow. One was going to open up cation channels. This one was linked to this this other cation, but going the opposite way because it's potassium, right? Remember, potassium was high on the inside and it flowed out. When this positively charged molecule leaves the cell becomes more negative. And you get this inhibitory response. All right, so same neurotransmitter, different effect based on the receptors that were involved right here. And we saw some similar stuff with the hormones a little bit. When you get to things like you're looking at the heart, the parasympathetic, sympathetic innervation of your heart, you'll see that, a lot of it's due to this, whether you cause a depolarization or hyperpolarization based on this different types of channel right here. So now we know that when we talk about the summation effects, that is trying to reach that threshold, trying to reach that minus 55 millivolts so that sodium channels would open. Right here, you have a lot of competing interests, right? Some of these are excitatory, talking in, and some are inhibitory. If your excitatory is greater than your inhibitory, and that excitatory is strong enough, you get an action potential. If the inhibitory is big enough, then you don't get an action. It's big enough to cancel these, and you don't get an action potential. What you often see in these neural circuits is that instead of having more excitatory input, you just kind of block these inhibitory. So all this was going on and there was no signal. And then all of a sudden you kind of block these over here. And then all of a sudden that made this whole thing more excitatory. And that was because another neuron, so this was the neuron over here, somebody else was inhibiting him. Right? So if you inhibit an inhibitor, you end up with this positive charge right here. Or sometimes the opposite happens. You might excite this one more and make it more inhibitory, right, over here, right? So these are very complex sort of interactions that is just continuously, constantly bouncing around in your brain and somehow doing all your crazy brain functions. Again, you could think of this as a concrete kind of example. If this was your neuron that was, telling you to eat the Ben and Jerry's ice cream, right? And you had your memory one saying how bad you felt after. You had this one saying don't, for whatever reason, your willpower, right? And then you had these ones you're saying, go, do it, right? And then if something kind of weakens these, that's when you go get the Ben and Jerry. That's what stress does to you. It just kind of weakens. If you use your willpower too much, you have to like continuously repress yourself. You get weak after a while, and then you start eating a lot of ice cream or whatever you do, taking drugs. Choose your thing. All right, that wasn't making any sense. All right, but in any case, when we talk about this neuron, this purple one firing, right? If you had that spatial summation, that is two of these kind of activating at once, right? Double the fun, and then it activates. Or this one talking a lot, go, right? And either way, you can reach summation. Then you also had, now we have that inhibitory response, which might cancel an excitatory one. And so these are different kind of ways of summation there. So that is how the decision of a neuron to fire based on the total sum of its input, excitatory, inhibitory, and how those excitatory and inhibitory graded potentials are created by the particular ion channels that were in the postsynaptic membrane. So next time we'll talk about some specific neurotransmitters and a little bit about neural circuits, and we'll see you next time.